Hello everyone, good afternoon. Welcome back to the session of enzymes. So here in this session we'll be learning about factors affecting the velocity of enzyme reaction or the factors influencing the enzyme activity. So we all know the binding of the substrate to its enzyme or enzyme to its substrate is the prerequisite or essential for the enzyme activity. So these are the different factors which influence the enzyme activity, that is your enzyme concentration, substrate concentration, temperature, hydrogen ion concentration or the pH of the media, product concentration, presence of activators, time, light and radiation. Okay, so let us see each of the factors, how they influence the enzyme activity. So first is your enzyme concentration. So the rate of the reaction or the velocity is always directly proportional to the enzyme concentration when you are providing the sufficient substrate for the reaction. So here the velocity of the reaction is always proportionately increased with the increase in the enzyme concentration, and again when you are providing the enough substrate or unlimited substrates. Okay, so when you plot this on the graph, so you will get a straight line curve here like this. On the y-axis you are plotting the velocity of the enzyme reaction, enzyme catalyzed reaction, and the enzyme concentration on the x-axis. So here you can see increasing the enzyme concentration is increasing the velocity proportionately. So this property is made use for the determining the level of particular enzyme, any of the enzyme in plasma, serum or the tissues for the diagnostic purpose. Okay, so next is effect of the substrate concentration. So here the rate of the enzyme catalyzed reaction will be increased as you increase the substrate concentration, and it increases until the certain point or until the maximum velocity is reached. So further increase in the substrate concentration after the maximal velocity, there will be no change in the velocity is observed. Okay, there will be no any effect once the maximum velocity is attained. Okay, so you will get the plateau or the flattened curve. Okay, so when you plot this you will get a rectangular hyperbola. So here you can observe, so initially when there is a less number of substrates and there is the velocity will be less, and further increase in the substrate, the 50% of the enzymes are attached with their substrate, and further increase in the substrate concentration, so all the enzymes are already occupied with their substrates, and here you can see the maximum velocity which is attained, and furthermore increase in the substrate is not changing anything, or there is no effect of further increase in the substrate when the enzyme concentration is kept constant. Here you are not increasing enzyme concentration, you are increasing only the substrate concentration. So furthermore increase in the substrate concentration after the maximum velocity is attained, there will be no effect in the reaction velocity. This is due to the complete saturation of the enzymes here. So this is how you will get the rectangular hyperbola curve when you plot the enzyme catalyzed reaction velocity versus the substrate concentration, and here we have kept the enzyme concentration constant, we are not increasing the enzyme concentration. So the maximum velocity, which is called as Vmax, and it represents the maximum reaction rate, and the half maximal velocity where the 50% of enzymes are attached with the substrate is called as Km. The Km is nothing but Michaelis-Mention Michaelis-Menten constant. Okay, so this is about the substrate concentration, and we have an equation called Michaelis-Menten equation. So this is the mathematical setup which express the rate of the reaction in the equation. Okay, so this will be written as V is equal to V max into substrate concentration divided by the Km plus the substrate concentration. So this Michaelis-Menten equation will explain or describes how the reaction velocity varies with the substrate concentration. So here you can see the relationship between the rate of reaction and substrate concentration. Okay, so here the V stands for the initial rate of the velocity, whereas S stands for the initial concentration of the substrate, whereas V max is the maximum velocity attained with the highest substrate concentration when all the enzymes are occupied or saturated, and the Km is called as Michaelis-Menten constant, and this is how you are going to write the equation. So I'm not going to explain about this Michaelis-Menten equation in detail, but we should know what is Km and what is its significance. Okay, let us see what is Km. So Michaelis-Menten constant or Michaelis constant, it is the constant or the substrate concentration which is required to produce half maximal velocity, that is half Vmax. Okay, so where the 50% of enzyme molecules are bound with their substrate molecules at the particular substrate concentration. So that is called as Km, and it is expressed in moles per liter. The Km value will be expressed in moles per liter. So this is your Km. Half of the Vmax or maximal velocity will be expressed as Km. So here the 50% velocity in the y-axis is extrapolated to the corresponding point in the x-axis. So that gives you a numerical value of your Km. So the Km is the signature of your enzyme. So these are the salient features and the significance of your Km. As I said, the Km is the signature of the enzyme, and it is constant for any enzyme. Okay, the Km value is constant for an enzyme, and this is a characteristic feature of the particular enzyme for specific substrate. So lesser the numerical value of your Km, higher the affinity of the enzyme to its substrate, whereas more the Km value of the enzyme will shows the less affinity for the substrate for its enzyme. Okay, so Km is always independent of enzyme concentration. Increasing the enzyme concentration will not increase or decrease the Km value. So here I have a example of glucokinase. The numerical value of Km of the glucokinase is 10 millimoles per liter, whereas hexokinase is 0.05 millimoles per liter. So this indicates the hexokinase is highly having a higher affinity towards its substrate, for example glucose or the hexoses, whereas the glucokinase has lesser affinity compared to the hexokinase. So 50% molecules of hexokinase are saturated even at a lower concentration of the glucose. So very minute amount of or minimum amount of glucose will be converted to glucose 6-phosphate by this hexokinase enzyme, because even minimum amount of glucose will be to say the hexokinase has a higher affinity, so the minimum amount of glucose can also will be converted to its product, that is glucose 6-phosphate, by the enzyme hexokinase. So that is how the hexokinase has a more affinity for the glucose than glucokinase. Okay, so because the hexokinase has very less Km value, that is 0.05 millimoles per liter. So coming to the effect of temperature. So the Km will be asked as a 2 or 3 marker question separately, and its significance. Okay, so coming to the effect of temperature. So the velocity of the enzyme reaction will always increase when the temperature of the media is increased, and it reaches the maximum. Then further increase in the temperature will reduce or decreases the enzyme catalyzed reaction. That means the velocity falls. So that is how you will get the bell shaped curve when you plot the catalyzed reaction versus your temperature. So the temperature at which the maximum amount of the substrate is converted to its product per unit time is called as optimum temperature of that particular enzyme. So all the enzyme has their own optimum temperature. So here you can see the bell shaped curve when you are plotting the reaction versus your temperature. So here the reaction velocity on the y-axis, whereas on the x-axis you are plotting the temperature. So here increasing the temperature is increasing the velocity, and further increasing the temperature will make fall of the velocity. So this is the particular temperature is called as optimum temperature of that particular enzyme. So at this temperature there will be maximum conversion of substrates into product will be observed. So when temperature is more than 50 degrees Celsius, there will be a heat denaturation and consequent loss of the tertiary structure of proteins, that is your enzyme structure will be lost. Hence the activity of the enzyme will be decreased. That is how the velocity will fall. So the most of the enzymes will of our human enzymes will be active in between 35 to 40 degrees Celsius, and they have optimum temperature in between 35 to 40 degrees Celsius. And whereas certain enzymes which has the optimum temperature near 100 degree Celsius, which are bacterial enzymes, for example Taq polymerase enzyme, which is active, okay, in the higher temperatures. So coming to the effect of pH. So increase in the hydrogen ion concentration also influences the enzyme activity. So each, as like your temperature, each enzyme has their optimum pH also. So in that optimum pH there will be a maximum velocity will be observed, and with, as I said, with few exceptions like pepsin which need a 1 to 2 pH, whereas ALP which is highly alkaline 9 to 10 pH, whereas acid phosphatase require 4 to 5 pH. All except these enzymes, all the other enzymes will be active with the optimum pH of around 6 to 2, that is they are active in our body pH. So on both the sides of which the velocity will be drastically reduced. If you increase or decrease the pH of the media, you will see the drastical reduction of your enzyme catalyzed reaction or the velocity. So when you plot the graph, it will show the bell shaped curve as like for your temperature. So the pH here in your enzymes decides the charge on the amino acid residues at their active site. The pH of the media will decide the charge on the amino acid residues at the active site. So the net charge on the enzyme protein would influence the substrate binding and the catalytic activity. That is why the pH is very important. So here you can see the activity is high, pepsin activity is high in between the 1 to 2 pH, whereas the acid phosphatase or amylase which is active between this pH where you can see here and arginase which is active in between 8 to 9 pH. So this is how the optimum pH, in that particular pH the activity will be maximum. Coming to the effect of product concentration. So the accumulation of the reaction products, that is your products, when the substrates are converted to products, there will be decrease in the enzyme velocity. The accumulation of the products will decrease the enzyme velocity. So as we know, there will be a feedback inhibition mechanism which will affect the enzyme catalyzed reaction. So for certain enzymes, the products combine with the active site of the enzyme so that they will form a loose complex. That is how they will inhibit enzyme activity. That is called feedback inhibition. So in our living system, this type of inhibition will be generally prevented by quick removal of the products formed. Okay, so that is about the product concentration. Coming to the effect of activators. So some of the enzymes require the activators or the inorganic metallic cations and the anions, that is your metalloenzymes or metal activated enzymes, right? So we have many activators which will activate the enzyme action or enzymes. Okay, so the cations which require for the enzyme activity are example magnesium, manganese, zinc, calcium, cobalt, copper, sodium, etc. Okay, for the activity of the enzymes, whereas anions like chloride which is required for the amylase activity. So we all know the metal which functions as a activators for the many of the enzymes. Okay, how the mechanism is, they may combine with the substrates or they may combine with the enzyme substrate complex and they will form a enzyme substrate metal complex, or they may directly participate in the reaction and bring some conformational change in the enzyme. So that is how they act or they influence the enzyme activity. And we have two different enzyme categories, that is, one is metal activated enzymes and metalloenzymes. The metal activated enzymes, where here the metal is not tightly bound to its enzyme protein. Okay, for example, the ATPase is an enzyme where it requires magnesium and calcium, and enolase which require a magnesium. Here the metal ion is not tightly bound with its protein or the enzyme, whereas metalloenzymes, where the cation or where any of the metal ion will be tightly bound with their enzyme proteins. Okay, so they are not readily exchangeable. So examples are the carbonic anhydrase, aldolase, alcohol dehydrogenase, which contain zinc as a part of them. Okay, and they are tightly bound with them. Phenol oxidase, which contain copper in them. Pyruvate oxidase, manganese. Xanthine oxidase contains molybdenum, and cytochrome oxidase, which require iron and copper, which are tightly bound with these proteins. Okay, so coming to the effect of time. So under ideal and optimum condition, the time required for the enzyme catalyzed reaction will be very less. So variation in the time of the reaction are generally related to the alterations in the pH and temperature. If you alter the pH and temperature, they may require more time or excess time for the catalyzed reaction. Okay, so usually the enzymes require very less time for the conversion of substrates into their products. And coming to effect of light and radiation, so exposure of these enzymes to any of the ultraviolet, beta or gamma or x-rays will inactivate them. Okay, so due to the formation of the peroxides. So for example, UV rays will inhibit your salivary amylase activity. So that is how the enzyme activity will be affected when you expose them to any of the light and radiation. So by this I'm finishing by the factors which will influence the enzyme catalyzed reaction. So for any queries you can contact on WhatsApp, and in the next session, later sessions, we'll discuss about the other enzyme kinetics in detail. So for today, thank you. Thanks for your patience.